Hello, everybody. My name is Itzhaky. I'm a CEO and co-founder of Maxonis, and I'm going to present Maxonis results to you in the next presentation. So, Maxonis breakthrough nerve regeneration is built on LDP-916. This is a novel peptide embedded within an injectable hydrogel for nerve regeneration application. We already were able to demonstrate robust functional recovery in preclinical models, complete and incomplete models. We are currently conducting safety studies to support first-in-human trials in 2027. And the company was founded by myself, Itzhaky, and Professor Shimon Rochkin, the world-leading expert in nerve regeneration. This is our team. Part of this team is working together for the last 25 years. We established two very successful tissue engineering companies that were able to get FDA approvals for its product and distribute the products all over the world for several medical applications. We also have a scientific advisory board, including Professor Eric Zager from the US, Mr. Jim Martucci, who is a major R&D manager in Baxter, and Professor Thomas Kretschner, who is also a world expert from Europe, as well as our Professor Shimon Rochkin. The base of our technology is a peptide, which is called LDP-916. It's based on laminin. Laminin is a structural extracellular matrix protein providing tissue scaffold, cell adhesion, migration, and maintenance. And it's a short 16 amino acids peptide, optimal for nerve regeneration. If you look at the drawing, this is the structure of the laminin. And we actually are taking two sides of the molecule and combine them together to create the LDP-916, which is our major innovation. We have a full plan of pipeline for R&D. As you can see, our main two programs are PNI, peripheral nerve injury, and spinal cord injury, where we finished the efficacy preclinical trials, and we are moving to safety preclinical trials in order to get to pilot human studies at the beginning of 2027. We are also inquiring traumatic brain injury and also other applications as well. This is our plan for the next 2 years. Actually, you can see they are divided into three major projects. The first project is the scale-up that we are doing to the technology. This is an injectable material, and we are developing it fully, including analytical and everything needed in order to get to a CMC position by the end of next year. We are doing the safety studies as we speak, moving from non-GLP studies to GLP studies according to FDA regulation. And we are discussing with the FDA and other regulatory authorities, doing pre-IND this year and next year until submission of the full IND before starting the clinical trials by the end of next year. So let's start with some data that I'm going to show you. And just for understanding, we have built a full array of preclinical trials from different angles in order to prove that our technology can treat in different situations the spinal cord injury situations. We are using two animals types, rats and mini pigs as well. And as you can see, we are using two types of spinal cord injury models, acute and chronic, using full resection and contusion in different applications, and also using direct injection during open surgery or direct intrathecal injection as well. So actually, we are doing the combination of all of that in order to prove and reassure that our results are valid. So let's take two examples. The first example is a complete acute spinal cord injury rat model. And when I'm talking about an acute model, I'm talking about full resection. And actually, we are taking 2 millimeters of the spinal cord out in order to make sure that the control group is not going to recover. So if you can look at the pictures from left to right, on the left side, you can see the spinal cord itself. Next to it, the resection of the 2 millimeters. Then we inject the material inside, and then we cover that with a membrane and wait for the results. And as we look at the BBB scale, we can see that indeed the control group is not moving and is very close to zero, even after 6 months. So there has not been any recovery in the rats. While the treated group, you can see that slowly but surely it's recovering until it gets to the point of the 6 months results. This is also supported by electrophysiology. You can see the motor measurement and the sensory measurement from left to right. And you can see clearly that there is a recovery on the motory, which is significant, and at the sensory as well. And this is coping with the BBB results as well. But this is a very severe model, and we wanted also to take a look of what is happening in a regular, incomplete, acute model for spinal cord injury. So in that case, we chosen the contusion model, as you can see, a 12 weeks follow-up for that. So again, we are injecting directly to the site in this case. And then we get, after 12 weeks, the results of the BBB, which of course is increasing compared to the 2 millimeter resection. But also we can see that the control group has some recovery, as with all the company results that we have seen so far. But when we look at the results of the group here, I think that we can see that the control group on one side, if we are breaking apart the BBB to 3 groups, until between 0 to 7, it's the early stage, 8 to 13 is the intermediate stage, and then the late stage is 14 to 21, when 21 is a full recovery. We can see that the control group is always in the early stage, all the 15 animals, while if we take a look at the 12 weeks, we can see that 4 of the animals are almost fully recovered, and 7 of the animals are partially recovered, and that gives us almost 80% of motor recovery, which is very impressive. If we look at the electrophysiology, it's coping with those results. 27, 28% recovery on the motory. And we can see that the sensory is getting very high score on 12 weeks. And that complete the recovery of the animals in this model. So if we move to the summary of what we have seen so far, so we have a breakthrough injectable therapy for nerve regeneration across application. It's a proven efficacy in extreme preclinical model and validated in multiple independent labs. This technology, based on peptide, it's safe, cell-free, and off-the-shelf solution. It has a strong IP portfolio, protected, innovative, and future pipeline. And the first in human trials are planned to be in 2027. So in conclude, I urge all of us, let's bring a real solution to millions living with untreated nerve injuries. I would like to thank everybody and thank you from the Maxonis team.